What if the most potent plant growth regulator, the most profound soil stimulant, and the most abundant source of plant hormones on Earth could be brewed in your backyard for mere pennies? Forget the expensive bottles lining garden center shelves, promising a revolution they can't deliver. Today, we're not just talking about making a simple brew. We're dissecting a sophisticated bioreactor that harnesses nature's most ancient and powerful chemistry. This isn't just about mixing ingredients, it's about orchestrating a microscopic revolution in a bucket. We'll explore the detailed biophysics, the nuanced chemistry, and the complex biological pathways that make this solution a living factory. We're going deep into the science that big agriculture hopes you never uncover, the intricate dance of microbes, starches, and minerals that can transform your garden, your farm, and your understanding of fertility. At the heart of this system lies a revelation that began quietly in South Korea, championed by farmers like Cho Han-kyu and his son Cho Young-song. They observed the untouched resilience of forest ecosystems and asked a simple yet profound question. If a forest can build its own fertility for free, why can't our farms? Their answer, distilled into a powerful technique known as JADAM Microbial Solution, or JMS, defies the industrial model of agriculture. It's a method for cultivating what they called Indigenous microorganisms, or IMO, right at home. But let's elevate this understanding beyond simple observation. What exactly makes a handful of forest soil, a potato, and some sea salt so astronomically powerful? It comes down to microbial metabolism and the exquisite engineering of nature. When you introduce soil microbes to a nutrient-rich environment, you're not just feeding them, you're triggering a cascade of biochemical synthesis that produces a veritable cocktail of plant growth-promoting substances. Consider the humble potato. Many recipes suggest simple sugars like molasses, but here's why complex starches, like those found in boiled potatoes, are a superior fuel for fostering robust, diverse local IMO guilds. Molasses provides readily available sucrose and glucose, leading to an immediate, explosive, but often short-lived microbial bloom. This rapid spike can favor fast-reproducing, often less diverse microbial species, creating a metabolic monoculture within your bucket. It's like feeding your microbes a sugar rush, intense but unsustainable. Potatoes, on the other hand, are rich in amylose and amylopectin, long-chain polysaccharide starches. When potatoes are boiled, these starches gelatinize, making them more accessible.
Then, the indigenous microorganisms, particularly various species of bacillus, pseudomonas, and fungi, produce extracellular enzymes like amylase. Amylase breaks down these complex starches into simpler sugars over time, like a slow-release fertilizer for your microbes. This sustained nutrient supply encourages a more stable, diverse, and robust microbial population, fostering guilds that work synergistically, mimicking the balanced ecosystems found in healthy soil. This slow fermentation promotes the growth of a broader spectrum of beneficial bacteria and fungi, leading to a richer array of plant growth regulators. Then there's the often underestimated role of sea salt. It's not just a mineral supplement. It's a critical osmotic stabilizer and a selective pressure agent. The human body maintains a precise saline balance, and so do microbial cells. When microbial cells are placed in an environment with vastly different solute concentrations, they face osmotic stress. Too low solute outside the cell, and water rushes in, causing the cell to swell and burst, a phenomenon known as osmotic lysis. Too high, and water rushes out, leading to dehydration. The roughly 0.1% concentration of unrefined sea salt in JMS creates a slightly hypertonic or isotonic environment for many beneficial soil microbes, reducing osmotic shock and supporting cell wall integrity. Furthermore, unrefined sea salt provides a broad spectrum of trace minerals, magnesium, calcium, potassium, sulfur, and dozens more, which are essential cofactors for countless enzymatic reactions within the microbial cells. These trace elements are often missing in highly refined tap water or even some filtered waters. Beyond osmotic regulation and nutrition, salt also acts as a competitive exclusion agent. At this precise concentration, it subtly inhibits the rapid proliferation of many common pathogenic bacteria, like Salmonella and E. coli, which often struggle in higher salinity environments or in the presence of robust lactic acid bacteria that thrive in fermenting conditions. This gives the beneficial, forest-derived IMO a competitive edge, ensuring a safer and more effective brew. Now, let's talk about the brewing process itself. It's a marvel of biochemical engineering, accessible to anyone. Here's the practical, step-by-step recipe for brewing your own powerful JMS. Step 1. Water preparation. Start with approximately 4 gallons of non-chlorinated, non-chloraminated water. Tap water is often treated with chlorine or chloramines, which are biocides designed to kill microorganisms. This is the exact opposite of what we want to do. If you only have tap water, fill your bucket and let it sit uncovered for 24 hours to allow chlorine to off-gas. For chloramines, which don't off-gas easily, you can add a small amount of ascorbic acid, vitamin C, or use a carbon filter. Rainwater or well water are ideal as they often contain naturally dissolved minerals beneficial for microbial growth. Step 2. Potato power. Take 1 pound of plain white or yellow potatoes, typically 2 to 3 medium-sized potatoes. Wash them thoroughly but do not peel them, as the skin contains additional beneficial microbes and compounds. Cut them into 2-inch chunks. Place these potato chunks into a pot with enough dechlorinated water to cover them, about half a gallon. Boil the potatoes until they are very soft, easily mashed with a fork. This boiling process gelatinizes the starches, making them highly bioavailable for microbial enzymatic breakdown. The starchy boiling water is invaluable. Keep it. Step 3. IMO collection. This is the crucial inoculation step. Venture into an undisturbed, healthy deciduous forest or woodland. Gently push aside the top layer of leaf litter and collect 1 to 2 cups of the dark, crumbly, earthy material found 2 to 4 inches beneath the surface. This is your leaf mold, teeming with an incredible diversity of indigenous microorganisms, including fungi, bacteria, and archaea. If a forest isn't accessible, use the most biologically rich, mature compost or worm castings you can find from a healthy system. This is your starter culture, the genetic blueprint for your powerful brew. Step 4. Assembly, the bioreactor construction. Take a clean, breathable mesh bag, a nut milk bag, a fine mesh laundry bag, or even a clean knee-high stocking works perfectly. Place all the boiled potato chunks into one mesh bag. In a separate mesh bag, place your collected leaf mold soil. Suspend both bags into your 4 gallons of dechlorinated water in a non-metallic, 5-gallon bucket. Metal buckets can react with the acidic fermentation byproducts, potentially inhibiting microbial activity. Now, carefully pour the starchy potato boiling water into the bucket as well. This adds an immediate carbohydrate boost and beneficial minerals. Step 5. Saline stabilization. Measure precisely 2 tablespoons of unrefined sea salt. This is approximately 30 grams, aiming for a 0.1% by weight solution in 4 gallons, approximately 15.1 liters, of water. Dissolve the sea salt thoroughly into the water in the bucket. Stir well. The salt is not just for minerals. It regulates osmotic pressure, protecting the microbial cells, and acts as a selective agent. Cover the bucket loosely with a lid or cloth. Do not seal it airtight, as fermentation will produce gases that need to escape. Place the bucket in a shaded area, ideally between 60 and 85 degrees Fahrenheit, 15 to 30 degrees Celsius. This temperature range optimizes microbial growth and activity. Step 6. The fermentation begins, observing the biophysical indicators. Within 12 to 24 hours, you'll observe the initial signs of life. Small bubbles may appear, indicating the metabolic activity of aerobic bacteria like Bacillus species, which rapidly multiply in the oxygenated water. By 48 hours, the surface of your brew will be covered with a thick, frothy, almost yeasty foam. This is a critical physical indicator of peak microbial density and vigorous respiration. This foam is primarily composed of carbon dioxide gas, trapped by microbial exopolysaccharides and cellular debris. The density and consistency of this foam are direct correlates to the exponential growth phase of your microbial colony. In warmer conditions, this foam might peak sooner. In cooler conditions, it might take 72 hours or slightly longer. When this thick, creamy cap forms, your JMS is alive and at its most potent. The microbial population has shifted, with facultative anaerobes and lactic acid bacteria beginning to dominate, producing beneficial organic acids that further solubilize minerals and inhibit potential pathogens. Application, precision, and strategy. Once your JMS is fully active, it's ready for deployment. This is where the true art and science of regenerative application come into play. Your JMS is a living elixir, not just a fertilizer. Dilution. Always dilute your JMS before application. A standard dilution ratio is 1 part JMS to 10 parts non-chlorinated water, 1:10. For very young or sensitive seedlings, or as an initial test, you might start with a more conservative 120 or 150 dilution. Never apply undiluted JMS directly to plants, as its acidity and high microbial load can cause osmotic stress or even burn tender tissues. Timing. Apply JMS during the early morning or late evening. This is when plant stomata, the tiny pores on leaves responsible for gas exchange, are typically open. Applying during these times maximizes foliar uptake of the phytohormones and microbial metabolites. Midday applications in hot sun can lead to rapid evaporation, reducing efficacy and potentially causing leaf burn if the solution dries too quickly. Branch testing. Before applying a new dilution ratio or a batch of JMS to an entire crop, perform a branch test. Select a small branch or a few leaves on a representative plant, spray or drench that section, and observe for 24 to 48 hours. Look for any signs of stress, yellowing, or adverse reactions. This allows you to fine-tune your application without risking your entire harvest. Crop stage targeting. Different plant growth stages benefit from different hormone profiles. During vegetative growth, auxins and cytokinins are key. During flowering and fruiting, brassinosteroids and gibberellins play a larger role. While JMS provides a broad spectrum, consistent application throughout the growing season ensures that plants receive a balanced hormonal boost. For intense vegetative growth, weekly applications are beneficial. As plants transition to flowering and fruiting, maintain a bi-weekly application. Apply as a soil drench around the root zone, or as a foliar spray directly onto leaves. Advanced customizations and considerations. The basic JMS recipe is incredibly powerful, but you can enhance it. Genetic drift risks in cereal inoculation. The practice of cereal inoculation, taking a portion of finished JMS to start a new batch, skipping the leaf mold, is a cornerstone of JADAM for continuous production. However, it's important to understand the concept of genetic drift. With each successive inoculation, there's a selective pressure that favors the fastest growing and most dominant microbial strains in your specific bucket environment. Over many generations, perhaps more than 5 to 10 batches, this can lead to a reduction in the overall diversity of your microbial community, as less competitive or slower-growing beneficial species may be outcompete. To counteract this, it's advisable to periodically restart your JMS from fresh leaf mold collected from the wild every few months. This reintroduces a full spectrum of local biodiversity, preventing your microbial guild from narrowing. Additional starches/inputs. Some advanced practitioners add a small handful of rice grains alongside the potatoes. Rice provides a different carbohydrate profile and can introduce additional beneficial starch-digesting bacteria. Others experiment with a small amount of roasted barley or other grains to broaden the nutrient base. However, always ensure the complex starch principle is maintained. Aeration. While JMS is typically a facultative anaerobic ferment, aerating the finished brew for the final 12 to 24 hours using a simple aquarium air pump can shift the microbial population towards more aerobic species. This can sometimes boost the production of specific compounds or increase the shelf life of the active solution, as aerobic microbes can be more stable. Rainwater advantage. Cho Young-sang consistently emphasized the use of rainwater. Beyond being free of chlorine, rainwater contains a unique profile of dissolved atmospheric gases and trace minerals from its journey through the atmosphere, which can subtly, yet significantly benefit microbial populations compared to filtered or well water. This isn't just about growing bigger plants. It's about restoring the fundamental biological integrity of your soil. Long-term practitioners report soils becoming darker, more friable, with vastly improved water retention and nutrient cycling. Plants exhibit enhanced disease resistance, reduced pest pressure, and often significantly increased yields. Most importantly, your garden becomes less reliant on external inputs over time, as the introduced microbes establish self-sustaining populations in the soil itself. However, it's crucial to acknowledge the limitations. JMS is not a magic bullet. It requires a baseline of organic matter in your soil. Microbes need carbon to feed on. Severely depleted soils will still benefit from compost and mulching. It's temperature sensitive. Below 60 degrees Fahrenheit, fermentation slows dramatically, and freezing temperatures will kill the active culture. Finished JMS also has a limited shelf life, typically 2 to 3 weeks at room temperature before the microbial population shifts towards putrefaction. Make it, use it, make more. This is a living product. The global biofertilizer market is a multi-billion-dollar industry, projected to exceed $8 billion by 2030. These commercial products often contain single, specific strains of bacteria, freeze-dried, and packaged for shelf stability. They are standardized, patented, and profitable, but they often lack the synergistic power and biodiversity of a wild, mixed microbial population. A bucket of homemade JMS, costing less than $2 per batch, provides a dynamic, diverse ecosystem of dozens, if not hundreds, of different microbial species working in concert, a complexity that cannot be replicated in a commercial bottle. This knowledge, once a linguistic and cultural blind spot, is now freely available. Farmers like Cho Han Kyu and his son Cho Young Sang refused to patent their discoveries, opting instead to share this profound science with the world, empowering individuals to take control of their own soil fertility. They showed us that the most powerful soil amendment isn't a proprietary blend or a patented chemical. It's the living, breathing biology beneath our feet. The science is clear. Indigenous microorganisms produce the exact plant growth regulators. Auxins, cytokinins, gibberellins, and even brassinosteroid analogs that scientists spent decades isolating and industries now sell for exorbitant prices. This biology is not fringe. It is the fundamental engine of every healthy ecosystem on Earth. It's available to you for the cost of a potato, a handful of forest soil, and 5 minutes of your time. Armed with this deep understanding of the biophysics and biochemistry, you're not just brewing a solution. You're cultivating an intelligent, self-replicating biological factory. You're reconnecting with an ancient wisdom, validated by modern science, and reclaiming your power to grow a vibrant, resilient garden. If this deep dive into the microscopic world changed how you view soil, fertility, and the potential in your own backyard, please subscribe and share this knowledge. Let's collectively empower ourselves and regenerate our world, one bucket at a time.